Today I am going to tell you about cancer and the modern targeted therapy used against this dangerous disease. As you know, cancer is a disease in which some of body's cells begin to grow and divide in uncontrollable way. Normally, cell division and cell death are strictly regulated processes, and any new cell appears only when it is really needed. But for some reason, this mechanism breaks down, and a tumor cell begins to divide over and over again, forming a tumor. To treat cancer, we need to stop this process of division and to kill tumor cells. But it is not an easy task, because cells are cells, and it is really challenging to kill a tumor cell, but not to make any harm to a normal cell. And that's the reason why all anti-tumor drugs are very toxic and cause severe side effects. However, recent decades, several approaches of targeted therapy have been developed which allow to reduce side effects. To understand these approaches, we need to consider the differences between normal and tumor cells. You can see on the picture that normal and tumor cells differ in terms of shape and size. Usually, tumor cells can be different in size, and their shapes is very irregular. A nucleus inside a tumor cell is usually larger compared with normal cell, and it contains a abnormal number of chromosomes. But what is the most important thing is that some of chemical reactions inside tumor cell are different comparing with normal cells. In human cells, chemical reactions usually go one by one, so that product of one of the reaction serves as a reactant or substrate for the next chemical reaction. And in this way, chemical reactions form a cascade or signaling pathway. And some of the signaling pathway leads to cell division. For example, here you can see some of this pathway which lead to cell division, and generally in normal cell, these pathways are inactive most of the time. But in tumor cells, due to some mutation, these pathways can be activated most of the times. How can we stop this signaling for cell to divide? So the most obvious idea is just to destroy or inactivate some chain of this cascade of reactions. For example, RAS protein, when it is activated, it binds to RAF protein, and as a result, a signal goes that cells should divide. If we destroy this interaction between RAS and RAF, no signal will go. You can see that in normal cell, RAS becomes activated only in presence of a growth factor. Then activated RAS binds with RAF and signal for cell division goes. When no growth factor is present, no activation occurs, so RAS and RAF don't interact with each other, and no cell division occurs. Due to some mutation in tumor cell, RAS can be activated all the time. So this binding between RAF and RAF proceeds over and over again. So the signal goes and the cell divides and divides and divides. How can we prevent this signaling? We can use some small molecule which binds, for example, with RAF protein and prevents RAF from binding with RAS protein. We can use small molecule which binds, for example, with RAF protein and prevents RAS and RAF proteins to bind to each other. In this case, no binding between RAS and RAF occurs, no signal goes, no division happens. This is one of the ways when we prevent cell from division. But there is another way. We may want just to kill tumor cell. How to do it? We can send some poison inside the tumor cell. But before entering tumor cell, our poison should travel throughout the whole body, throughout the bloodstream, to do it safely. The scientists use the strategy of the Trojan Horse. Let me remind you the legend. The war between Greeks and Troy lasted for many years. And in spite of a long siege of Troy, Greeks could not conquer the city. So they thought of a trick. They built giant wooden horse, and 50 of their best, strongest warriors were hidden inside this horse. Then Greeks pretended to sail away and left the horse on the shore. When Trojan citizens saw that Greeks had gone, they were very happy. And then they noticed the horse on the shore, and on the side it was written that this is the gift to the Poseidon god from the living Greeks. So they didn't want to make Poseidon angry, opened the gate and brought the Troy inside the city. But the same night, warriors got off the horse and killed the guard and army and conquered the city. So probably we can use the same strategy to bring our poison inside tumor cell. The thing is that some receptors are overexpressed on the cancer cells. So it means that the concentration of these receptors on the surface of tumor cell is thousands of times higher compared with normal cells. And we can use these receptors as a gates to bring our poison inside the tumor cell. And here on this picture you can see some of these receptors. But let's consider one more time why the strategy of Trojan horse was successful for Greeks. First of all, the horse seemed harmless to Trojan citizens, so they didn't suspect any trick and brought the horse inside the city. The second factor is that the warriors were very strong and very fearful. And the third factor is that all the warriors were locked inside the horse until the horse got inside the Troy. So we can use the same strategies, the same three factors to make our chimeric molecule successful. First, our horse, our carrier, should be as natural as possible. So tumor must not suspect any trick. We need to use natural proteins or antibodies or natural peptides. Second, our warrior's drug must be very active and very potent and should be able to kill the cell even in very low concentration. And the third and very important thing, our linker should be stable during circulation and release drug only inside the tumor cell. And this approach appeared to be very successful in medicinal chemistry. So now there are several drugs when antibodies are used as a horse. So these antibody drug conjugates, Milatarg and Besponsa, are used against leukemia. Trodelvy and Kadcyla can be used against breast cancer, Elahere against ovarian cancer, and Zynlonta against lymphoma. But as you know, not only chemotherapeutics, but radiation can be used against cancer. But usually, when we apply radiation to the whole human body, it's very harmful. We can use the same approach and attach our radioactive stuff to some horse which bring radioactive stuff inside the tumor cell. And this approach was realized in Lutathera, the drug which is intended for diagnostic and treatment of neuroendocrine tumors. In low concentration, our radioactive stuff can be used for diagnostics. So under X-ray radiation, the surgeon see where the tumor is located, and so he can do surgery more precisely. In higher concentration, this radionuclide peptide conjugate can be used to treat cancer. The same idea is realized in Pluvicto, the compound which is used for diagnostic and for treatment of prostate cancer. So you can see that the idea, the strategy of the Trojan horse can be used to conquer the city, but also it can be successfully used to win the war against cancer.